Hi there. On our last video, we talked about innate immunity in length. This time, on this video, we will shift our focus to adaptive branch of the immune system. And let's start with something that we are already familiar with. You might remember this listing of the two branches of the immune system from our earlier discussion. And of course, this time, our focus will be on the adaptive branch. So let's discuss it in a little more detail. However, before we get started, I need to once again give a warning. This is a huge topic and there is no way that we could cover this in a sufficient detail that I feel that is needed to really understand it. So we will just do our best to have a general overview of it. That sounds like a plan to me. Sometimes this branch of the immune system is described as acquired immunity, and I think that it is a rather good term to describe it. So the idea here is that an individual gets infected once and then builds an immune memory, which we call immunity in a layman's term, so that if this individual meets this pathogen again, there is already a plan ready to how to handle this pathogen. So one could say that there is a memory of this from the past. This system is made of highly specialized cells and processes. So here is a really nice diagram showing the innate immunity on the left and adaptive immunity on the right. And we also see the different cells that are involved in each of these. Also, please note that there is overlap between the two, just like I said in our very first video when I introduced you to the immune system. But at large, if we were having to characterize or compare and contrast each in some way, one could say that innate immunity is more general, so it is not as specific, but it acts much faster. While adaptive immunity is specific, so really hitting the target with an appropriate response. However, it is slower. So in our last video, we discussed how innate immunity had all these lines of defense, making physical barriers like skin and mucous membrane, chemical protective layers like pH, sweat, and oils, and then of course also cell response. This time, we will shift our focus on the adaptive branch. So let's see what we can explore with this branch. So, as we have seen already earlier, the adaptive immune system branch is a very specific defense system, which can eliminate almost any pathogen or abnormal cell in the body. It does this by amplifying the immune response and activating the complement. However, the shortcoming of this branch is that the system must be primed. This means that we need to have had an initial exposure to a specific foreign substance before this system becomes effective in responding to it. And this priming, as we call it, takes time. And there are many different ways how one may become primed to a specific pathogen, and we will discuss them in a more detail later. But some examples would be, of course, having an infection or a vaccination where we are exposed to a small dose of inactivated form of the pathogen. Or if we have a baby, it can receive mother's antibodies, for example, from breast milk or even earlier through umbilical cord. So what are the benefits of this system? Well, it is very specific, so it recognizes the target and acts on it. It is also systemic, and by this I mean that it is not limited to the initial site. And finally, it generates a memory. This means that if we get a second exposure to the same pathogen, there is a very strong attack against it, as the antigen is already known to us. So at this point, I would normally play or sing Memories by Barbra Streisand, but in the interest of not making this uncomfortable for you and me not getting into trouble for using her song, I will skip that. Just know that the memory aspect of this is very important in this branch. And we can divide adaptive immunity to two further branches. Humoral branch uses antibodies which circulate free in the body fluids. When they find an invader, they bind to it. And this is where B cells are going to be important for us. Cell-mediated branch acts through lymphocytes, the white blood cells in the lymphatic system. It can either act directly, which refers to killing the infected cell or calling other lymphocytes to deal with the pathogen, so we can think of this as if it would tag the pathogen with a sign, "Kill me." And now we are looking at the T cells in this system. I have provided this rather nice review of these two in your learning system for you to study in your own time in more detail. But now I think it is important for us to familiarize ourselves with some terminology. This is largely just revision of what you already probably know to start with. Antigens were foreign particles that enter the body. They should not be there. They do not belong there. And they are likely to cause trouble if they get to spread and multiply numbers within the body. So essentially, antigens are the target of our immune response. They are something foreign which we must get rid of. Antibodies, instead, are the workhorse of the immune system. They circulate within the body, and when they find a pathogen that should not be there, they recognize it and possibly may also act on it. And often you might hear these being referred as immunoglobulins, IG for short. And some of our testing relies on seeing if we have formed these immunoglobulins against something because we have faced this pathogen. You can think of, for example, COVID-19 antibody testing. This is what we are looking for in these tests. Have we already faced COVID-19? Note that it does not tell if you currently have it, just if you have met COVID-19 virus in the past. If you wish to consider this visually, I have provided an imaginary model of each here. Notice how antigen, the pathogen in this case, so something that should not be in the body is gray. And antibody, they are what protects your body, circulating within it and trying to detect if there's something foreign. These are bluish. And it is like a lock and key mechanism. The antigen and antibody must be a match. Your textbook seems to suggest that antibodies are always Y-shaped. But please note that this is not always the case. In reality, we have many other shapes. Now I would like to shift our focus on something different. Let's discuss about 3 important cell types that we see in the adaptive immune system. So we have lymphocytes. These are our B cells and T cells. Remember, these were associated either with humoral immunity, and this was B cells, or cellular immunity, this was T cells. In addition, we have antigen-presenting cells. These are often abbreviated as APCs for short. They play an assistant role, showing an antigen to other immune system cells. We will start with looking at B and T cells. And what we are going to do next is to describe the development, maturation, and activation of the B and T cells. So essentially, we are looking at their entire life cycle. Note that both B and T cells share the same steps, so you only need to learn it once. I know that this might seem a little theoretical, but it is important for us to know this process. Your textbook offers some nice illustrations that you might wish to use to complement this discussion. Have a look from there for illustrations that might be helpful. So let's work through what happens. Initially, both B and T cells precursors originate in the red bone marrow. B and T cells mature in the primary lymphoid organs, and this is bone marrow for B cells and thymus for T cells. During this time, these cells achieve something that is called immunocompetence. This refers to the lymphocyte being able to recognize only one specific type of antigen. Remember, there were many different kinds, and antibodies needed to match these exactly, like a lock and key mechanism. So, when B and T cells have matured, they now only display one type of receptors for antigens on their surface. They can only bind to that kind of antibody. Another important thing that happens is that these cells gain self-tolerance. This means that they are unresponsive to one's own antibodies. So your B and T cells are not going to attack you. Only foreign invaders are deemed as pathogen. That makes sense. This is quite important, right? This maturation process includes quite a few steps during which these cells that fail to have immunocompetence or self-tolerance, they are deleted. And those who are very interested in this topic, I would not expect everyone to memorize this part. I could share that we actually know the maturation process of T cells pretty well, but it is somewhat less understood when it comes to B cells. I guess that the take-home message here is that the B and T cells go through an education during which we make sure that they mature to what we want them to be, and if they fail to be that, they get eliminated. So a little like an internal quality control process, almost. Okay, what happens next? Well, now these B and T cells are ready to travel to secondary lymphoid organs. Remember these from our last chapter. So any B and T cells that are not yet exposed to antigen are called native. So as these B and T cells travel from primary to secondary lymphoid organs, within the circulation, there is a chance of an encounter with an antigen. Well, if this encounter happens, this causes the B or T cells to continue its development, and this would include these cells multiplying in numbers and becoming activated. And this means that they have two possible futures. They can either act on an invader. This would be what would cause an infection. Or they can become memory cells These cells are important as they stick around in the immune system in case the same antigen is met again later, so that the immune system is able to act quickly at that time. This is what we often think of becoming primed or prepared to a pathogen. We have a template against it based on our previous encounter. In layman's terms, this is what we think when we talk about immunity to something, right? So that is the life cycle of B and T cells. And this is all I need you to know about B and T cells at this level. So that was our B and T cells. Now we should look at the antigen-presenting cells. They do what the name suggests. When an APC meets an antigen, it cannot act on it itself, but it alerts the rest of the immune system, almost like, hey, there is an antigen here. So they are basically helpers. And there are different types of antigen-presenting cells: dendritic, macrophages, and B cells. I will leave covering these for your own interest using the textbook if you are curious to find out more about this. So we have seen this slide before. If we wish to recap, humoral immunity, which is shown on the right, was all about being caused by the antibodies. And in cell-mediated immunity, which is shown on the left, there was no antibody involved, but this was activated in response to antigen. You will have this diagram as a handout in your learning management system, so feel free to check it out from there. I think that it is a rather good one. Now I think that we are ready to wrap up this video here. So in our very last video for this unit, which you can find after this one, we will talk about primary and secondary immune responses and also some more about relevant clinical relevance. And this is a hot topic now, I know, so maybe it will help to explain a little that too. I will see you in that video.